Welcome to another Postdoc Career Talk webinar. For those who don't know me, my name is Lorena. I'm a postdoc at FAU at the Brain Institute, and I am the current president of the Postdoc Association, and I will be the host for today's talk. So before I introduce our speaker and we move on with this webinar, I just want to give you a few quick announcements. If this is your first time here, or if you're not very familiar with this series, this is a collaboration between FAU's Postdoc Association and the Office of Postdoctoral Affairs. And this is to serve our postdoc community. We're bringing PhDs from different fields and career paths to share their journey with us, share their experience in their daily activities or how they got there, and some tips that can be valuable for us. So we know that FAU has postdocs in many different fields, so we are trying to acknowledge and bring speakers that can match all of that. So stay tuned, and each month we're gonna have a different talk. And if you have any special requests or a specific career that you're interested in, just let us know. Any feedback that you have is appreciated, so just let us know. Just one more quick thing, since we are on a webinar format, you are not going to have your videos and your microphones turned on. So, but we want to hear from you, and we want to address all of your questions that you have to our speakers. So please use the Q&A button, this green button at the bottom, type in your question there, and I will redirect them to Dr. Lee. So I'll try to bring up the questions as they come and turn this as interactive as possible. So if you have a question regarding something that he's speaking about right at that specific point, just let us know and I'll try to bring it up. And any remaining questions that we have will be addressed at the end. So without further ado, it is a pleasure to introduce Dr. Sooyoung Lee. He has an extensive experience in the pharmaceutical industry, working with biopharmaceuticals, and he's had leadership roles in many companies, and he is the founder and CEO of the company called Cellucis here in Florida. So welcome, Dr. Lee, and thank you very much for sharing your time with us.
My pleasure to share my experience as a biochemical engineer for the past 30 plus years in biopharmaceutical industry. And I started my career as a chemical engineer after getting my BS. I worked in the refinery for several years before I decided that, hey, biotechnology is something new coming up. And so I went to graduate school to get my Ph.D. in biochemical engineering. And I'll be happy to share my experience, and feel free to ask questions, I guess, at the end of the discussion. Yes. So this diagram, can you see it?
Yes, we can.
Yeah, this diagram is actually first blockbuster biopharmaceutical EPO, as you know, and then this is a protein and the receptor as a diagram. I'll be talking about the evolution of biopharm industry, how we started, great promise of genetic engineering, followed the Human Genome Project. Was the promise fulfilled as of now? And discuss a little bit about biopharm industry landscapes. What are the participants and what opportunities there might be in biopharm industry? And we'll talk about unique challenges in biopharm industry, especially that this is a highly regulated industry and you need to receive an approval from FDA, which make it more challenging, as well as the challenges in manufacturing biopharmaceuticals, which is what biochemical engineers do. So the modern biotechnology with great promise, as you all know, started in 1953 with Watson and Crick and deciphering CDA, double helix of DNA. And then, of course, then that prompt people searching for the application of DNA technology. There are many options at the time, DNA for forensic techniques, DNA to modify crops and agricultural use, DNA-based industrial enzyme, pharmaceuticals, vitamins, as well as DNA-based drugs and insulin, growth hormones, EPO, as we all know. And then, of course, there is also DNA-based diagnostics. So really what prompted the DNA promise is that in 1973 is the first time Professor Cohen and Boyer at Stanford established genetic engineering and demonstrated the feasibility of transferring gene from one organism to another. And then there are another 1977 is the first successful transfer of genes from human genes to E. coli, microbial E. coli bacteria. And that prompted great interest in biopharm industry. When you talk about biopharm, and when you talk about biopharm and therapeutic proteins, basically we're talking about, actually first biopharm, defined as biotech-derived pharmaceuticals, was started in 1796, Edward Jenner, when he developed the smallpox vaccines. So until 1982, most of the biopharm or biologics were called, were the vaccines. But in 1982, United States, FDA approved recombinant DNA protein insulin. And this is the first time that recombinant DNA protein was manufactured in large scale by inserting human genes in E. coli, and the first time a DNA-derived therapeutics were approved. So the big question is biotech is a promising industry? A lot of people are questioning that at the time. But more importantly, biotechnology is the technology searching for applications. When you think about biotechnology, it's a technology, it's not an industry. It's like electricity, right? The electricity is a technology, but you need a lot of application of electricity. Same thing happens here. So they are searching for applications, and it could be agricultural, therapeutics, and it could be other diagnostics and other areas. And the following event actually prompted, and a lot of focus was focused on biopharmaceuticals, because early on 1985, there was some success in 1980s, 85, the new vaccine was developed by small biotech company startup, Praxis Biologics. And this is at the University of Rochester. Professor Smith developed biotech company. And actually, vaccine industry was being dominated by giant pharmaceutical companies at the point. So this is the first time actually they do biotech farm is approved and become a catalyst for major changes in biopharm industry. And then followed with 1986, the recombinant hepatitis B vaccine was developed by Merck. And then in 1989, this is very important milestone because Epogen is one of the first blockbuster drugs. It defined the annual sales of $1 billion. And that was developed by healthy kidneys to make red blood cells by recombinant therapeutic technology there. So then, since then, the rush for biopharm discovery was actually fueled by a human genome project. Basically, as you all know, it's a mapping chemical sequence of human DNA. And it was hailed as the first great technological trump of 21st century, or even our lifetime, if not of the history of mankind. And it just started in 1990 and was declared complete in April of 2003. And this is a result of advanced biotechnology techniques and science, as well as supercomputers. And I think Dr. Venter, who was part of the Human Genome Project, said what once took 10 years can now be done in 15 seconds with the aid of supercomputers. So this advances in biotechnology and application of computer technology, sharing information, and other aspects continue to increase the number of new drugs being developed dramatically at this time. This diagram shows the increase in genomic data over time. In 1980, 589 times, they're only 10 megabytes. And right now, 10 gigabytes and terabyte, I guess. Right now we're talking about 20 petabytes. So there's tremendous increase in knowledge on human genome, and that will prompt the discovery of new drugs that's coming out at this time. So what's promised realized? When you look at this diagram, and actually this is the number of drugs approved in 2014, in 2017. I don't have data beyond that at this time, but basically increased from 34 to over 60. And when the number of the drugs from a period of 1989, there was small numbers, and as of the last 4 years, there were 112 approved. So you clearly see the increase and dramatic increase in biopharmaceuticals, and actually becoming I think the promise is being kept at this time. Interesting to note is that out of all these drugs approved the last 4.5 years, there are 68 monoclonal antibodies, 23 hormones, and 16 clotting factors, and so on. So clearly, monoclonal antibody is a favorite of the technology, and that's what prompted the revolution of biopharmaceuticals. Second revolution, I should say, the discovery of monoclonal antibodies and utilization of them. So, in short, the FDA approved 59 drugs in 2018, of which 39 was biotech-derived pharmaceuticals. It used to be that this number was much less than a few in 1980s. So clearly, the industry has adopted biopharmaceutical in a major focus in biotech industry. And I briefly mentioned dominance of monoclonal antibodies. Again, approval as a percentage of drugs approved, and you can see that right now it's up to 53%, and then it's followed by the amount of revenue generated. So this list shows the top-selling prescription drugs by revenue in 2019. It's 10. The red are the biopharmaceuticals. The other one is small molecules, so traditional drugs, the chemical entities. You clearly see that, and every single one of these top 10 antibodies is denoted by MAB, monoclonal antibodies. Even this embryo is monoclonal antibody as well. So it shows that the BioPharm is here, and it is no longer a promising industry, but it is a great industry to be at this time. I'll give you a little background of the unique challenges and aspect of the biopharm industry. The 20th century, I guess 21st century, is actually a century of IT and BT. The major difference, I guess the similarity is that they are highly technically driven industry. However, major difference is that BT is regulatory driven. It needs to be approved by the federal government to be able to market. Whereas IT, you can actually start the business, and within 2 or 3 years, you can become a billion-dollar industry. Whereas in BT, will take 10 to 15 years before you realize the fruit of your technology. So it's highly technology driven, it's challenging discovery process to discover new drugs. And also it requires complex manufacturing process because we are really utilizing living animal cells, if you will, and genetically engineered. If you think about the cells, their main mission in their life is to replicate, to continue. While you are doing by doing a genetically modification of the cells, you are forcing cells to produce something they normally don't produce. And as you know, cells do not listen to people that well. They got mind of their own. So that's a major challenge that we have to face with. It's regulatory driven in a sense that they must prove the safety and efficacy through human clinical trials. And oftentimes you have to follow good clinical practices by law. And of course, you need extensive quality system to meet the regulatory requirement compliance for manufacturing and testing of these things to define as a good laboratory practice, GLP, or good manufacturing practice. These are by law that you need to follow and comply with to be able to produce biopharmaceuticals. It's very risky in a sense that only 40 in 2000, and this number can vary depending on which way you look at, which articles you look at. Basically, can be as much as 2,000 to 10,000 number of promising candidates. And then there will only small fraction, maybe 40 or even less, the product candidate can enter clinical trial, human clinical trial. And the success rate is 13% through phase 1. And there are 3 phases of clinical trial, basically, phase 1, phase 2, and phase 3. And some of them are 13%, 35%, 59%. As time progresses, the chance of success is greater. But again, this percentage can vary depending on the nature of the product and the complexity of the project. However, the pharmaceuticals chance of approval is somewhat higher. Therefore, it's really resource intensive for product development. High cost of development, about $1 billion. Again, it can change from $1 billion as high as $2.5 billion for the drug. It depends on what it is. Just look at the magnitude of it. I cannot count $1 billion, as you know. And the long-term investment, it takes about 10 to 15 years before people promise the candidate you identify become a reality and the drug approved. This diagram shows the ecosystem biopharmaceutical development. As I mentioned, that a lot of recombinant DNA technology utilized to discover new drugs. You identify a protein of interest or antibody or whatever, hormones and enzymes, and then you discover that one and make sure that it is potentially, it can potentially cure, excuse me, human disease. Then you follow with preclinical development. This is where you do a lot of animal testing and make sure that it's safe and it shows some efficacy. You have to use an animal model because you don't want to test it on human being up front. And you know, animals are not a good model for human disease. As a matter of fact, my old boss used to tell me, we make wonderful drugs for mice. Actually, a lot of drugs that are tested for mice never make it to clinical trial area. And then you enter, this is what we call development area. So that is preclinical, as where tox study, animal study is done. And you develop manufacturing process for production of clinical trials. Again, the challenge here, excuse me, is that when you, for clinical trial, you need to make sure that it's safe. It means that you need to make sure that it's made safe and it can be reproducible. And therefore, the cost of experimental drug can be as high as $1 million for those, if you will, because you need to make it as if it's commercial. What the chance of failure is very high, as I mentioned, for clinical trial, but nevertheless, you need to do this. So this is one of the critical areas in development. Once FDA approve it, then you go to large-scale commercial manufacturing, and then you follow the marketing and distribution. All of these, each components are very important because even if the drug is approved, there are cases where approved drugs after 10, 15 years and spending $1 billion or so can fail in marketing, and that is not unknown. So you need to make sure that you do all these things equally well and based on the risk and the resources you require. In order for this infrastructure ecosystem survive, you need to have all this support system at the same time. There need to be R&D center, biotech ventures. There will be animal testing laboratories. CTO is a contract testing organization specializing in testing, very complex testing. CRO is a contract research organization that works with the hospitals and clinical trial aspect of it. CMO is a contract manufacturing organization that does a lot of clinical manufacturing, and there will be a large manufacturing facilities. So you need sometimes a manufacturing facility that be as big as 1 million square foot. The reason is that you wanted to make it so that all the product, all the each vial of billions doses are consistent and same. So you want to make it in one place where everything can be made and uniformly. And that's why sometimes they have large clusters of large manufacturing facility in Ireland and Puerto Rico, Singapore, and these are some of the areas that manufacturing is taking place. And of course, that to support this activity, you need to make sure that you have adequate suppliers of raw material, consumable equipment, engineering services to make this happen. And marketing is important, logistics, distribution centers, the marketing sales office is equally important to make the drug a success. And the reason is that though some of these therapeutics, the proteins are very labile, and you know to make sure that they are kept at the right temperature, and you cannot, for example, shaking this drug sometimes can cause a lot of problems with the drug quality. So all this need a lot of science-based practice to make this happen. That's why in the United States, there are certain clusters like San Francisco, Boston area, North Carolina, Washington, Baltimore area, maybe even San Diego area has biotech cluster in there. Unfortunately, Florida, we don't have one at this time, but you need a lot to support the infrastructure together as a cluster to support all these activities.
Dr. Lee?
Yeah.
Mind if I interrupt for a second? We have a couple of questions that just fit with what you were just talking about.
Right.
One of them is if the biopharmaceutical companies, do they usually manage the clinical trials themselves, or do they have to hire or partner a company that is specialized in the clinical trials?
Yeah, good question. Basically, it depends. If you're a large pharmaceutical company, you may have internal resources to do that. But many small biotech companies rely on CRO, which I showed on the diagram before. They're just specialized in clinical trial capabilities and work with hospitals to manage this clinical trial area as well.
Okay. And the other one is, do biopharmaceuticals like the antibodies and all of those drugs you just mentioned, are companies usually specialized in these biopharmaceuticals versus like just chemical compounds, or the bigger pharmaceutical companies usually work with both?
Yeah, I can talk about that a little in the next slide, but awesome. Yeah, I think this is the slide I can show you. Oh, yeah. So this is what the reason I put that up here is partnership. It will make this biopharm industry such fascinating and challenging because you cannot do everything yourself. You need to have certain specialties that people need to help you to work together. So biopharm industry, as I mentioned, start up as biotech R&D, university, government lab, R&D centers. The other one who discover potential drugs, and then you become a small biotech companies. There will be some, but actually the forefront of this biotech industry is those scientists whose mission is to discover the truth, discover the drugs, not for to making a lot of money, but for the sake of science. And that's what makes the biotech industry so rewarding for some people, and as well as that's what makes US become a best place for biotech industry. But some of them become entrepreneurial and they create a small biotech companies. And then they grow to become a big biopharma, such as Amgen, Genentech, and Biogen, all the names you hear. And then there's a big pharma. Big pharmas have traditionally been chemistry-driven small molecule companies. They don't have entrepreneurship and they don't have stomach for risk-taking. So therefore, they wait until this small biotech comes to the biotech. This drug is ready to enter clinical trial. Then that's where they would form a partnership and to create a partnership to proceed further. And there are service providers I mentioned. CMO is people who are manufacturing, because manufacturing these drugs requires special technology, and that's when an area that I have certain expertise in it to follow to do that. And small companies cannot afford to have that. Only big companies can do it, or their organization that specialize in producing it under GMP conditions for clinical trial as well as commercial. The CRO is what we talked about, and CTO is a testing company that specializes in testing. And then there will be a partnership being formed, and then another partnership in big pharma and big biopharmaceutical companies. You can see that certain drugs are co-developed. Eli Lilly have collaboration with Pfizer, and they have a marketing right in Europe versus the US. So there's a lot of collaboration take place between the pharma because nobody can do it by yourself, right? So this is the infrastructure of how the biopharm industry developed. More importantly, this may answer your question. Basically, majority of new drugs approved originated at or were initially developed by small biotech companies. When you look at this diagram, number of approvals in 2018, 63% of that from small biotech companies, whereas only 9%, over 9% is from big pharma companies and big pharmaceutical companies. And there are other larger biopharmaceutical companies and mid-sized biopharmaceutical companies that are small. So the majority of the new drugs are being developed and originated at small biotech companies. At a certain point, they will form a joint venture or collaboration with the big pharma companies in a way. That's the that's the nature of biopharm industry in a sense. This diagram may tell you the reason why that happened. You know, we mentioned that typical diagram drug development approval process diagram, discovery, we talked about that, a preclinical where animal study is done, and clinical phase 1, phase 2, and phase 3 clinical trial in human being, and then the FDA review process. This whole thing takes about 10 to 15 years. Again, don't specifically, there's each drug is unique. It may take 8 years, it may take 20 years, depending on what it is. The millions, I put down about $1 billion, but again, the number can vary from billion to 2.5 billion or even higher. The important thing is the clinical trial takes about half of the cost, whereas discovery is $100 million, or 1/5 of that. So many small biotech companies are focused on discovery. Once they enter in preclinical, it's a little more expensive, and they will enter clinical trial, they have a choice of raising more money or have a collaboration with the pharmaceutical companies. Pharmaceutical companies do not mind paying a little more because they don't have to take the risk up front. But once they enter clinical trial, the chance is a little higher, so they are willing to invest at that time. That's how the ecosystem works, partnership works in there. So number of drugs, again, it can be 4,000, 10,000, depending on who is counting. And you enter maybe animal study, but that narrow down to about 40. And then in the clinical trial, and maybe 5, and you to get 1 drugs out of there. This is the process, what is unique about the biopharmaceutical industry. So I just recently saw this article, cost of drug development have been published, and an estimated 2.6 is, I think, 2014 number. But this is exaggerated, I think. On the average, about $1 billion right now. But according to this article, it was 1990s, so 1.4 billion. 1980s is $400 million. So this ever increasing cost of drug development, and depends on how you account for this development cost. This number can vary 2 to 3-fold. But important thing is the magnitude of investment and time and resources that you need to put in to make this happen. Talk about a little bit about clinical trial, because really clinical trial is the gatekeeper of biopharm industry. Phase 1 is initial safety testing in a small group of healthy volunteers, 100 or maybe 30 to 40 to less than 100. And the main goal is to demonstrate it's safe. It doesn't kill people. You test it on healthy volunteers. When successful, you enter it phase 2, and the success rate is 14% for given drug, but it can be as high as 25%. It depends on what drug it is. Phase 2 is assess safety as well as efficacy in a small group of patients, typically somewhere between 100 to 500 patients. And there you start looking at different dosage. You don't know how much you should give and dosage regimes. Once they look successful, 35% of that goes into, phase 3. And this is a pivotal study. It demonstrates safety as well as efficacy in a large group of patients, somewhere between 1,000 to 5,000 patients. It can be done globally, and this is where it's costliest and longest trial. And it requires comparison hundreds of studies in many hospitals around the globe, because certain drugs respond differently based on racial makeup, DNA sequence of the individuals. So there are a lot of factors you need to look at, and this is major. But once you enter phase 3, the chance of success is pretty high because you have gone through phase 1 and phase 2. So let's talk about the manufacturing process technology. So once you have two streams, one is that trying to receive approval from FDA. The other, you got getting ready for success and how we're gonna make it. When you think about it, FDA does not want to approve drugs that you cannot provide globally. You're talking about 1 billion doses a year, perhaps. And you need to make sure they can be produced in a larger scale, consistently, and each vial is almost same or equivalent to each other. That's a major task. So you need to be prepared to be able to produce it to meet this requirement. And there are several components of process technology. One is the expression system. They're basically biology. So the engine of all these biopharmaceutical is a cell line, recombinant cell line. Okay, that is being developed by most of the molecular biologists, cell biologists, and you need to characterize it from protein. There's a biological technology aspect of it. Production system with a process technology involved. So you need to be able to grow these cells, the large vat, large bioreactors, or you can even use animals, or you can use a plant to produce. You heard about edible vaccines, and you can transplant genetically modified bananas for the drugs as well. And then of course you need to recover and purify this. This is important because you know the technology advances that 10 years ago you're making a gram per liter of protein. You're doing wonderful monoclonal antibody I'm talking about. Right now, maybe the best one is as high as 5 grams per liter. But when you think about 5 grams per liter, that's what percentage? It's not even 1%, right? It's 5 grams per liter is a 0.5%. In other words, the 99.5% is water and media. So you need to purify this protein, very precious protein, expensive protein, costs $1 million or more to be able to purify and make sure that your purity is over 95% or more. So that means that you need to purify it, and sometimes you actually end up losing a lot of product because in the process of purifying, you lose about 50% of protein you make. So this is very, very important components of the process technology, which is not addressed early on, but later on as you enter, before entering clinical trial, you need to address this as well. Another important thing people do not think about is the characterization, the analytical technology. Modern technology and FDA is requiring more and more testing of the product to ensure that there is no impurity. As I mentioned that the purity should be 95%. When you think about it, 5% you don't know what you have in there, and you have no idea what that do to your health of a human being. So that you need to be characterized a lot of impurities and protein to make sure they're consistent and they're safe. And we're talking about in biologics, there's an axiom in biologics. That is, process is the product. How you make it, how to grow cells, actually is the product in itself. You cannot just test the end product because they're complex, large molecules, a lot of glycosylation patterns and other things that affects its functionality. So these are the key things that, and you cannot transfer one process and do it to another site. You need to demonstrate that they are equivalent. And you can use a different bioreactors, different purification techniques can make a huge difference. So developing biopharm manufacturing process is, as I mentioned, many biopharm drug candidates during R&D discovery phase on a bench scale. It's relatively simple to do that. However, bench scale process must undergo process development phase. And basically, you want to develop process that is reproducible, scalable, and robust process. This is key thing you need to do. They need to be reproducible. It can be done here. It can be in Asia. It can be in Ireland. And it can be scalable. That you can make it in a 5-liter scale, but you need to be able to do it in a 20,000 liter scale. And that has to be robust, means that you cannot have so sensitive the pH control cannot be just 7.05. You can maybe vary from 6.8 to 7.2. And you want to make sure that it does not affect product quality. And this is very important because that's why the federal government, they have a GMP guidelines that you need to comply with to maybe have to produce a large manufacturing scale, not only for commercial, but actually for clinical trial as well. So this process development area actually requires an interdisciplinary team of process scientists and engineers. Life scientists, microbiology, cell biology, molecular biology need to understand the cells they're working with much better, but physical, chemical science in terms of chemistry, biochemistry, physics, and for testing and understanding the purification process and all that aspect. As well as engineers like myself, chemical engineers, mechanical engineers, that you can need to scale up, and then IT and all this are put together. So when it comes down to, you need a interdisciplinary team to understand each other. The terminology people use between life science and engineers are different. Good example is that, when you talk about viral vaccine, people use virologists to use terminology of substrate, and they call animal cells a substrate because this is a virus use the cells to produce virus. For the people who are trying to grow cells using terminology substrate turns you off because this is something that we value. So, and then understanding the magnitude in the virology, for example, you measure virus in terms of log. Oh yeah, it produces about 5 logs of virus because there's so many means of the virus relatively simpler to measure, and the variability of virus population is measured by logs. Whereas in chemical engineers, the log of difference of 1 means a tenfold difference. So if we use a 5-liter bioreactor versus 50-liter bioreactor, cost of producing will be so much different. So you need to understand each other, and you have a common goal to develop this process together. This is an area that I think still being developed, and that there is certainly between this discipline, there is certain terminology and understanding what we are trying to do. But at the end, I think we all everything is based on science, and that's the key, the fun of this process development science. So we talked about GMP. Simply put, it's GMP are established to and maintained to assure the quality, efficacy, and safety of drug you're making, in a consistent manner. So FDA current requirement of GMP, requires, this is a regulation. By law, you need to follow this, and you need to define how the building, facility, organization, and personnel, who is accountable for what, the proper equipment and analytical equipment, control of component and drug. Sometimes raw material is very critical. There are, in all your cases before FDA established this criteria, white powder was used, which was, instead of sugar, it's a toxic material and killed a lot of people. So you want to make sure that all the components and raw material used meet the FDA guidelines. Production and process control, we talked about how we control the cell culture process, temperature, CO2 concentration, agitation, and other things as well. You need to make sure that packaging and labeling control is proper. How you label a given drug, if you buy any drug, there is a label, and that is very critical. How you label your drug can mean it's a failure or successful drug, because you need to list all the potential side effects, and therefore FDA is very careful how you to label it, and how to take it. Dosage level becomes very important. This is a very critical component as well. Holding and distribution of the drug is, as I mentioned, they tend to be pharmaceutical, biopharmaceuticals, very labeled, very sensitive to temperature and pH, and sometimes agitation itself can cause a lot of problems of aggregation of protein and things like that. Laboratory control is important because you need to be able to test the drugs as we go along. Test not only for the drugs, but also impurities and others, such as endotoxin, host cell proteins, and other things. And most importantly, the record and reports. Everything needs to be documented. Unless it's documented, it's not being done. That's a major thing about GMP FDA regulations. And actually, if FDA comes for an inspection, first thing they want to see is the documents. You want to make sure everything is clearly defined, accountability, responsibility, who does what, when did what, everything. As a matter of fact, in GMP, there are 3-level check. One is you define what you're gonna do, you verify you've done what you did during the operation, At the end, you review that you did what you're supposed to do. The three-letter check that documented, and everybody needs to sign it. And if you are in compliance, it really results in jail terms. And therefore, this is a very serious nature. And then, of course, you need to account for the return and salvage of product, because you need to make sure that the drugs are not diverted to illegal use, for example. So you need to make sure that all the drugs you produce are accounted for. So,
Dr. Lee?
yes?
Just one more quick question. So considering all these different roles that is involved in the whole process, right?
Right.
For a PhD that is transitioning from academia to the
Right.
industry, what do you think it's more important, their field of study or their skills? Let's say, for example, a technique or an equipment that they are familiar with. So for example, let's say someone is does a lot of cell culture and cloning and a lot of like molecular biology techniques, but was on a neuroscience program, for example. Would they have a chance applying to a company that doesn't work with neuroscience at all, but maybe that those skills could be useful?
Absolutely. I'll address that very quickly, actually, in the next slide. Actually.
Okay, awesome.
So when you talk about, you mentioned R&D and tox with animal studies and phase 1 and phase 2, you have to decide when you're going to do what. And these are some explanation of what things need to be done. Process description, version control of documentation, calibration, certification of TSC, raw material qualified, impurity studies. All these things need to be done, and you need to make a decision when you're gonna do it, because obviously you can do all this upfront, but why? Because you may not go through until phase 3. You need to make a proper science-based, and this is a key thing, science-based justification of what you do. A lot of this need to be done. You can be doing R&D phase, but there's no sense. You may not go to phase 3, but you can wait until phase 3, then it's too late. So these are the key decisions you need to make. So this is when should we do what is a major issue. Now, to answer your question, let me give you an example of to give you a little background on the manufacturing, and then maybe you can kind of get a sense. I know that I'm running a little late here, but bioreactor, and you go through purification process that we discussed. And this is, bench scale, and you see a bunch of cells. You are familiar, some of you may be familiar with how you grow cells, animal cells, these microcarrier attachment cells. But then you need to go through this manufacturing process, bioreactor purification, bioreactor in a large scale, purification is how you purify chromatic columns to remove impurities and other things as well. So, typical biopharmaceutical manufacturing organization, for that matter, I think discovery organization, but I want to give you an example of what other things that people need to do and what skill sets do we need. Obviously, there's the manufacturing science technology group that are doing a process development, right? And then they are the one who actually scale up and understand the cell line, understand why you're making all this stuff. There's quality control testing. So this will require biologists and engineers. Quality control testing, okay, biochemistry, HPLC, mass spec, ELISA, all that stuff required. Quality assurance is understanding all the techniques and make sure that it's done properly. There will be an engineering facility maintenance, engineering components to make sure the facility is in good shape. The manufacturing operation, those people, operators who actually operate manufacturing. There's the material management, the testing, making sure that raw material comes in and documented. Everything needs to be documented. When it comes in, you need to, the chain of custody is very important, and the project management to make it. Based on this, what do you think is most critical components of this activity, all of this? I would say the project management is one of the major challenges, because you have so many moving pieces. You need to do it in time. You need to do it appropriately. You need to understand it. To answer your question, I'm a firm believer in after you get your degree, you will learn everything you need to know after 5 years when you start working on the job. That's the area where you can learn. But the basis of your career is just science-based. Everything we do is based on science. And the even quality assurance, you're gonna ask why you're doing it. So it doesn't matter, as long as you have a good scientific background, you can always adapt to do other things. For example, a good friend of mine is a virologist. We work together, and he's a process scientist for GSK, or director of manufacturing. So it really doesn't matter what your background is. I think what they do in PhD, hopefully, I mean, this is the way I look at it, is that it teaches you how to think, how to problem solving, right? So sometimes it's more important to define the problem properly. It's 50% of solution, providing a solution. So you need to apply your scientific technology. Of course, you have to learn a lot of things, but nobody knows everything, as you know. I don't know anything. I'm not an expert in quality assurance, but you need to understand science behind it. You can always challenge people based on scientific, and unless you can understand it, you keep on asking, you will learn. So learning never stops. That's what I'm trying to say. So of course, if you have expertise in let's say how to do a cell culture, you naturally gravitate toward the natural cell culture, but do not limit yourself. Because what you do with the product when you do cell culture, why are you doing it? And what are the areas that affect what you do? Because it's not just growing cells, it's growing cells for the purpose of making a drug. And what components do you need to understand to make it successful? So you can do your job better. So I hope that answers your question a little bit.
Yes, it does. Thank you.
Yeah. And the last slide, this is everybody talking about COVID-19, right? This is right now, they say the 140 potential candidate vaccine. You think 19 will go into phase 1, it's about what, 10%, a little less than 10, more than 10%. We'll go into phase 3, 13 of them, phase 4, and then we'll have 1 approval. Now, they're saying you can do this in a year. That's different than 10 years, right? Vaccines sometimes can take a little shorter period of time, but this tells you that the way to go, see, one of the challenges the biopharm industry is take too long for the approval process. And this pandemic will teach us there are ways to shorten this timeline, reduce the cost. The way to do it right now, I think they are doing this right now, is just doing it concurrently. When you're at preclinical, you're getting ready for phase 1, phase 2, phase 3. Taking a risk, if it doesn't work out, I'm sorry, you lost it. But unless you are prepared for it, you're ready for it, so you can immediately go. It's not only from an industry point of view, but also from a government point of view. FDA is overwhelmed and to inspect, because there are 2,000 to 4,000 biotech companies globally, and everybody think they have the best thing since the sliced bread. I mean, they think this will cure all the cancer, but the FDA need to review them and you go to the next level. So I think there's a way, there's a means, there's a way to solve a problem, as we can see right now. Hopefully, we can have vaccine within less than a year, but that shows that it can be done. It's just a matter of where do you put the resources, how you strategize it. That's why I think it's very important that you, as a scientist, not only your techniques, but also how you can improve the process. Because all of us, I have been an observer and participant in this field, but I have not reaped the benefit of multi-billion-dollar company. But also the benefit, I think, we can all say, is that we help to cure the people disease and develop new medicine. So in the end, I think that's where we need to focus on ourselves. I think that's all I can say right now. Thank you for listening.
Awesome. Thank you very much. This was great. It's really big picture and also detailed. It really showed us how the whole system works, and that was great. Your last slide actually just answered a question. So someone asked, okay, it takes 5 to 15. So how are they doing this so fast for COVID, right? So you just answered that. That's great. Another question is, considering the amount of drugs that you listed in a graph, the proportion of drugs that are discovered or developed in the small companies versus the big companies, would you think that the market for PhDs, at least those that are more interested in the research and development side of things, a better market would be the small to mid-sized companies versus on the big pharma, or not necessarily?
Not necessarily, because I think when you think about it, the biopharma is only 7%, but that doesn't mean there are a lot of researchers you need to get to 7 approval. As you can see that, right? There's plenty of jobs out there. And I personally think, small pharma versus larger pharma. I worked in both cases. I worked for Schering and Pfizer, big companies, as well as small biotech companies as well. But the important thing, each has the pros and cons. I think working for larger pharmaceutical companies, it will help you to understand the process a little better. When you're for small companies, you are it sometimes. And so you need to be a risk taker, and so perhaps learning a little bit more about the overall picture in a big company is not. No, I think that's contradictory. What I'm saying in terms of science, large company may be able to provide a little better because you have mentors and there's a lot of people who have experience in this. However, big companies, they tend to pigeonhole in certain areas. So you can do a discovery in molecular biology. You're a molecular biologist for the rest of your career there, oftentimes. Whereas in a small biotech companies, not only do you have to do that, but sometimes you have to be a cell culture person. Sometimes you have to purify. So you have a lot of learning experience in there. So I think personally, you cannot be wrong either way. The important thing is that you have to learn. Nobody's going to teach you. And I think what the PhD does is it allow you to define the problem yourself and find the solution to the challenges. And you got to use your learning how to do it. And don't get me wrong, I think basically bioscience, what we do is all science-based, and you need to justify what you're doing there. So I'm sorry, I cannot give you one answer or another, but it depends on you. Some people like stability, some people like challenges, right? So risk versus reward, right? So that's what it is.
Yeah, no, that's great. It's a good discussion. I'm sure that's not a black and white
right.
answer. So that's great. So we have just one more question. And now for the audience, if you have any questions, just type them in right now, please, real quick before we finish. But one more question that I have here on the Q&A is if you could go back in time and talk to yourself right after you got your PhD, is there a specific or some advice that you would give yourself?
Well, I think I would have gone to work with Genentech. Then I would have become much better. No, I think it's a journey, right? It's I believe in wherever you are, you do your best, and I think there's a I love this movie called Accidental Tourist, right? I mean, everybody is a tourist, but it happened to be where you do. So I do not have any regret in there as well. My regret is that sometimes I took things for granted. Sometimes, that's a key thing. So you always make sure that why you're doing it, right? So you don't fall into routines, because then you become a routine person, right? If you want to be innovative and use your science, and I think you need to constantly explore. And if you are a cell culture person, if you're a molecular biologist, don't just stop there. What are they going to do with this thing, what I do, right? And then you ask questions, learn it, whether by talking to your colleagues or whatever. So, hopefully that gives you an answer. But I think the most important thing is that I learned all I know within 5 years of getting my PhD. And after that, I don't learn anything new. It's just application of what you know, application of methodology, how to define the problem and how to solve the problem, meet the challenge.
Okay, all right. I don't see any other questions right now, so I think we can wrap up here. Thank you so much again. This was great. It was fantastic. Wish all the best for your company. If you're ever hiring, you can.
It's a virtual two people operation at this time. So,
Okay, yeah,
okay.
I'm just saying, you know, we can help advertise it if you ever have positions available.
It's my pleasure to be able to discuss this. Unfortunately, I cannot get feedback, and I don't know whether too much detail on other things, but it's been fun. Thank you.
Okay, thank you. Bye, everyone.